Okay, Juan, get started.
Yeah. Okay, let's get started. First, I'd like to thank Rams for organizing this ZOOMinar series. This has been fun for everybody, at least to me. Okay. All right. So it's my great pleasure to introduce Matt Chapman, who is the first speaker today. Okay. Matt received his PhD in microbiology from Indiana University, and then did his postdoc with Scott Hotgren at the WashU in St. Louis. He joined my department at the University of Michigan in 2003. And in the last 18 years, he excellence in all aspects as a faculty member, including research, teaching, service, and was promoted to the full rank. Okay, so Matt is a true pioneer in protein folding and amyloid biology. So he raised the functional amyloid hypothesis in the field, which corrected the misconception that all amyloids are bad. Okay, he used the bacterial Curli proteins as a model system and studied their assembly function, cross-seeding with the pathological amyloids found in human diseases. And lately, he's trying to use bacterial Curli proteins or the properties of which he learned from his research to manipulate the aggregation and toxicity of the human toxic proteins. So Matt is well respected in the field. He serves as editorial editors or editorial boards of several journals, including like JBC, Journal Bacteriology, and several others. And he reviews grants from NIH, NSF, many other grant agencies, organize a number of international meetings, such as the FOSTA meeting on molecular mechanisms and protein aggregation. So he has about 100 publications and was invited to present his work in many places all over the world. So I would like to take this opportunity to thank Matt. Matt is my hero, friend, and supervisor in my department. So some of you may know that my department was not very nice to junior faculty, years ago. So I came to Michigan 2 years later than him. So his lab is just adjacent to mine, a few meters away. And he is the one which, I come to. So when I came to Michigan, he invited me to his house. And so I felt much welcomed here, much less nervous than before. So whenever I have questions, I always stop by his lab, his office, ask all the questions, including scientific and non-scientific. And so after we moved to the new building, he is in different tower, different floors. So I miss him so much. Matt, I miss you, okay? I'm so happy to see you here, and I look forward to your talk. All right, welcome, Matt.
Thanks, Wang. I need to hire you to give all of my introductions. That was very touching. Thanks. So I would just like to start off by thanking the organizers too. I know other speakers. I just want to echo the same things that I've heard over and over again. How nice it's been during these times to have this Saturday morning respite where we can get together and hear about some great science. And so hopefully I won't drive it off the rails today, and we'll keep this great series going throughout the summer. So today I'm going to talk about a story about polymerizing the fiber between functional amyloids and pathogenic neurodegenerative disease. I'm going to start by backing up just a little bit and talking about bacterial biofilms. So I'm a microbiologist at heart, and I've studied bacteria really my whole career and how they interact with their environment and with each other. And they do so in these communities, very ordered, structured communities called biofilms. And you can find biofilms all over out in nature. And the key point for today's talk and the rest of the time is that these biofilms are encased in an extracellular matrix that helps protect the cells that are underneath and part of the biofilm. And that extracellular matrix, in many, many cases, and really almost all cases where we look hard enough, contains at least one functional amyloid protein. And so, for example, the E. coli system that we studied, and we've studied for a long time, this uropathogen E. coli strain, has two major components in its extracellular matrix, both biological polymers that are really stable. One is an amyloid fiber called Curli, and the other one is a cellulose polymer, a glucose polymer. These two polymers interact with each other, intertwine with each other. They're both incredibly stable on their own. They have slightly different properties on their own. Together, they make a great house to protect your cells. And you can see here E. coli cells, which are protected in this biofilm by this extracellular matrix that you can kind of see as this spongy stuff on the outside of the cells. And so that amyloid component is called Curli. Curli are extracellular fibers assembled by many enteric bacteria, maybe all of them at least have the genes that we can tell. They're important for biofilm development. They help protect the cells from stressors. They're potent immune modulators. For today's talk, the important thing is that the major subunit of Curli is the CSGA protein, and the CSGA protein or Curli fibers themselves have many amyloid-like characteristics. So the first of them, when you look at these fibers on the cell surface of E. coli, they look like amyloids to people that have studied these fibers for a long time. And so you've got these straight, unbranched fibrils that emanate from the cell surface of the bacteria. If you take these fibers and purify them away from the bacteria and then study their biochemical properties a little bit, you find that they're extremely stable, just like other amyloids are. So you have to treat with something like formic acid in order to liberate the monomers away from these fibers. So they're really, really stable. And they have the cross-beta strand structure that's associated with all amyloids. And so here is a CSGA molecule. And you can see this cross-beta strand structure where you've got these repeats of these strand motifs, and then the individual subunits just stack on top of one another to build these fibers. These fibers bind to things like IoT and Congo red. So you can actually put Congo red into the plate of a growing E. coli colony. And if they're making Curli fibers, they will turn red, as seen over here on the left-hand side of this plate. Or if you make a mutation in that CSGA protein so that you no longer make the amyloid, they just look the normal kind of white color. They don't bind to Congo red. And so when we step back a little bit and think about amyloids, most of us have thought for many, many decades and really centuries now about how amyloid is sort of a foe, that you've got all of these proteins running around in our cells, and these proteins, are going to start out in some disordered state just by virtue of being synthesized. Most proteins will get into their native fold and stay in their native fold. Of course, some proteins never make it into their native fold, and they misfold once in their native fold, and they end up in some sort of misfolded conformation. Now, many of these can, of course, be chaperoned back onto pathway or degraded. That happens most of the time. But every once in a while, you get these aggregates which are very stable and or very persistent in their formation. And one of the most recognized of these is, of course, the amyloids. Now, in these cases where the amyloid is the product of protein misfolding, it's, of course, associated with all these diseases that we're familiar with: Alzheimer's, Parkinson's, Huntington's disease, and many, many others. But of course, that's not the only way to end up in an amyloid. You don't have to go through a misfolded pathway. There are many, many examples now of what we would call natively folded proteins. This is the fold that the protein is trying to get into, is this amyloid-like fold. And you could actually make an argument that the amyloid fold is really a persistent fold in protein biology. And these functional amyloids, these amyloids that are folding on purpose or forming on purpose, provide a lot of different physiological functions. And I've had a slide that looks like this for almost decades now, but certainly years, many, many years. And the first question up at the top, are functional amyloids the rule or the exception out there in cellular biology? And this was a completely rhetorical question when I first made this slide. There were many more examples of disease-associated amyloids, amyloids that had formed as the product of protein misfolding or not as a part of a dedicated pathway. But I think now there are arguably maybe more examples of these functional amyloids. Certainly bacteria make a ton of them. You're going to hear a lot about Curli today. You're going to hear about FAPC from Daniel later on, and there's many, many other of these bacterial functional amyloids. Yeast and fungi, even mammalian cells and animal cells will make functional amyloid proteins. Really, you can find functional amyloid proteins now in all walks of cellular life and in great numbers. This is a predominant thing in the cell. It's not just a one-off, I don't think. And so we think that these dedicated amyloid pathways hold a lot of promise for understanding how to control amyloid formation, how to control the timing and the placement and the efficiency. If you're going to build an amyloid fiber, you have to be cognizant as a cell how to balance those different potential toxic intermediates that we know can sometimes form. And so really the fundamental question that my lab has asked and is continuing to ask is, how do functional amyloids polymerize at the correct time in the correct place? And I think, you know, this picture kind of pulls it all together. Here's an E. coli cell. You're going to make these highly aggregation-prone amylogenic proteins in the cytoplasm of the cell, secrete them across a couple of membranes in the case of E. coli, and then assemble them on the outside of the cell into these beautiful amyloid sort of network on the outside. So how do you coordinate that whole process? This slide's got a lot of information on it. I only put it up because this is kind of the thing that my lab works on and has worked on for many years. And I'm not going to talk very much more about it, which is basically the biogenesis of these amyloids from E. coli's point of view. I want to focus you on two things. So we've got the cytoplasm of the cell down here. There's many genes that are involved in amyloid formation in E. coli. Some of those genes are nicely compartmentalized into two operons that are shown here, the BAC operon and the DFG operon. And I'm going to focus you in on this A protein. This is the major subunit of the Curli fibers. It's secreted through the inner membrane or cytoplasmic membrane. And then it needs to be secreted through this outer membrane, which is the second membrane in gram-negative bacteria. And CSGA gets to the outside of the cell. And so I think a remarkable thing about this protein is that it is secreted from the cell as a disordered protein. It doesn't have, as far as we can tell, any appreciable structure. It is mostly disordered. And then on the cell surface, it interacts with the growing fiber tip and forms these amyloid fibers with really quite remarkable specificity and quite remarkable efficiency on the cell surface. There's no ATP out there, there's no anything. So this amyloid fold is a perfect fold to work on the outside of the cell. And so we can do some biochemistry of this amyloid protein and study it both in a test tube. We've obviously got a great in vivo genetic system in E. coli to work on. But this in vitro system is equally as powerful. And so I told you that we can take the secretion machinery and we can take our CSGA protein. And if we overexpress our CSGA protein and the secretion machinery, we can get CSGA, which is secreted to the outside of the cell. And it is intrinsically disordered. It is not in an amyloid form. We can actually purify that protein. It stays relatively soluble for a little while. We have to work quite quickly because it wants to relatively quickly assemble into these amyloid-like fibers in vitro. And you can measure this by TEM, shown up here at the top of this slide, or by one of the standard thioflavin T assays. And just to orient you on these thio T assays, because I'll be showing you a few, we've got freshly purified CSGA protein at what we'll call time 0. This is right when we can get it out of a denaturing purification protocol. It is intrinsically disordered. no appreciable structure at this point. And just over the course of a couple of hours, you start to see that it will bind to thioflavin T and kind of go into this exponential phase and then into a stationary phase. So during this exponential phase, we start to see these fibers to form, and then something, either we run out of subunits or this fiber formation just gets a little bit less efficient as the fibers get longer and longer, and eventually we'll reach this stationary phase. And so when we've been studying this for years and what might modulate CSGA amyloid formation, both in vivo inside the cell and in vitro. And we've identified a few chaperone-like proteins that were sort of hand delivered to us by the Curli operons that I showed you before, those two operons, one of which was the CSGE protein. We had shown a few years ago that the CSGE protein in vitro and in vivo, we had a little bit of evidence, could keep CSGA in an unfolded or non-amylogenic conformation when you mix the two together in a test tube. This was a 1-to-1 stoichiometric ratio. You needed one CSGE for every CSGA in order to get this inhibition that we see here. And then we also studied another chaperone-like protein called CSGC. And this CSGC, I'm going to tell you just a short story about. This is older work, but I think it provides the link to get us to this alpha-synuclein protein. And so CSGC, here's the structure of CSGC. This was solved by Steve Matthews' lab. And about, well, I mean, the paper came out in 2011. So probably over 10 years ago now, Steve Matthews contacted me and said, hey, we've got CSGC. We're pretty close to getting the structure of CSGC. You want to collaborate, and your lab can do some of the biology and kind of figure out what this protein is doing. And I said, well, sure. You know, we hadn't studied CSGC very much because whenever we tried, we couldn't figure out what it was doing. But we thought with the structure, maybe we get farther. If you look at this 2011 paper, it's really nice crystallography work. Steve Matthews' lab did a great job. And then our part of the paper is a whole bunch of negative results. We don't know what CSGC was doing at this point. Luckily, I had a really brilliant graduate student named Maggie Evans. She was working on a different project and somehow genetically had stumbled on this. I've got a telephone ringing. I'm visiting my mom's house, and so everything is a little bit foreign. I'm sorry. Okay, there. So what Maggie had genetically stumbled on the possibility that CSGC was working as an anti-amyloid in the periplasm of E. coli. We had a whole bunch of the CSGC protein in the freezer, and so we just took it out and mixed it with CSGA in these DIO-T assays, and I think a pretty remarkable result. So here is CSGA forming amyloid quite quickly in vitro. And if you spike in a little bit of CSGC, and it doesn't take much at all, so you can do 1-to-100 ratio, 1-to-500 ratio, or 1-to-1,000, meaning one CSGC protein for 1,000 CSGA proteins, or 500, et cetera, et cetera. And you can see that at 1-to-100 and 1-to-500, you can completely inhibit CSGA amyloid formation. This is without ATP. This is without a energy source. This is a pretty remarkable finding, I think, or at least we thought it was. If you start to look around, so then we really wanted to say, oh, if you're going to build a functional amyloid, you got to have a CSGC. This is the thing that keeps these functional amyloids from forming in the periplasm. And then we kind of looked around in genomes, and we could find CSGA proteins in genomes of bacteria that didn't apparently have a CSGC protein. And it wasn't until we started to look a little bit more closely at this protein that Daniel's lab, I believe, annotated as CSGH, because it didn't have obvious sequence homology with CSGC from E. coli and from the gamma proteobacters. And but the CSGH protein was sort of in the same place in the operons, and it sort of looked like maybe it was a CSGC-like protein. So we solved the structure of that, modeled it against CSGC. I think you can see that it's a very similar looking protein. If you mix CSGH with CSGA, you can get the same sort of inhibition that we would see with CSGC. And so these really are similar proteins. So they really, there really is one of these chaperones for every time that you have a CSGA-like protein in the cell. Well, we were interested in the CSGC as maybe a general anti-amyloid protein. Maybe it was acting as some sort of surfactant or doing something to just generally inhibit amyloid formation. And so we poked around with a whole bunch of different amyloids. And most of the time, CSGC did nothing with them and had really no anti-amyloid activity, with one exception. And that one exception is the alpha-synuclein protein. And so I think this group is really familiar with alpha-synuclein, but it's associated with Parkinson's disease. It's an intrinsically disordered protein that forms amyloid. It's associated with membranes. We had some old evidence that some chemical chaperones had cross-reactivity with CSGA and alpha-synuclein, and this CSGC protein does also. So it also works as an anti-amyloid to alpha-synuclein. So alpha-synuclein gets into an amyloid. It takes a little bit longer in our hands, so it takes it 25 or so hours to start to form amyloid. And if you spike in a little bit of CSGC, here's a 1:100 ratio of CSGC. You can see you've delayed the onset of amyloid formation. And if you spike in a 1:10 ratio, you completely inhibit amyloid formation and really stick alpha-synuclein into a non-toxic, these small oligomers, which when we mixed with cells don't appear to be toxic. And so this kind of brought us to a question, is there a structural or biological connection between alpha-synuclein and bacterial amyloids? And I'll try and go through this story relatively quickly so that we can move on to Daniel's talk. So just a little backdrop on Parkinson's disease. Remember, Parkinson's disease is linked to the amyloidogenic aggregation of alpha-synuclein, severe motor dysfunction. Non-motor symptoms, interestingly enough, precede the motor symptoms by years, sometimes decades. And nearly 80% of patients suffer from constipation or some sort of gastrointestinal distress. And so when you talk to physicians, they will tell you this over and over again. And so the early onset of gastrointestinal dysfunction in Parkinson's disease suggests maybe a potential gut-brain link. And there's been lots and lots of work done on this in the decades before. And so alpha-synuclein fibril aggregates can migrate from the gut brainstem via the vagal nerve up to the brain. So if they're seated in the gut, they can actually move up that vagus nerve up into the brain mainstem. Brock's hypothesis was that some pathogen or some product of a pathogen in the gut could be responsible for the initiation of sporadic PD. And then 4 or 5 years ago now, Sarkis Mazanian's lab at Caltech and Tim Sampson in his lab had a really beautiful paper that really said that in a nutshell that the human gut microbiota from Parkinson's patients can induce motor dysfunction in a mouse model. So they had an overexpressing mouse model. And if they changed out the microbiome in those mice and gave them certain microbiomes from Parkinson's patients, they would have an increase existence of the alpha-synuclein in the amyloid-like form. And we wondered, could bacterial amyloids play any role in this? Could they be part of this? And so we collaborated with Sarkis's lab and Tim Sampson, who's now moved on to Emory. And what they did, and what we have is 2 different mice. We have wild-type mice and alpha-synuclein overexpressing mice. And into these mice, we either put Curli expressing bacteria that could make Curli, had the Curli genes, or bacteria which were missing the Curli genes and could not make Curli. And we just asked, what are the Parkinson's-like pathologies in these mice? And so in these mice, if you have alpha-synuclein overexpressed and the Curli bacteria colonized into their gut, so they're originally germ-free, we colonized these Curli-expressing bacteria in there, and you look at time to cross a beam, time to descend a beam, or time to remove a little sticky note that you put on their nose, you can see that all of those factors which are associated with Parkinson's disease in these mouse model systems are increased when you have both Curli-expressing bacteria and alpha-synuclein overexpressed. And so Curli-expressing bacteria by themselves is not good enough. And if you have mice that are not overexpressing or not able to make Curli, it doesn't matter if you have alpha-synuclein overexpressed. You still get the same phenotypes as a wild-type mouse. You can look at alpha-synuclein, actually aggregates of alpha-synuclein in the brain of these mice, and see that when you have wild-type bacteria that can express Curli, there's much more protease resistant and amyloid or punctate amyloid looking aggregates in the brain than there are with the CSGA minus bacteria. And so a question that came out of this is, could CSGA directly cross-seed or somehow accelerate the aggregation of alpha-synuclein? And there we just turn to the simple in vitro reaction where we have alpha-synuclein, which aggregates over a period of time in the Thio-T assay. And now we're going to spike in just a little bit of CSGA. So we're going to spike in a little bit of CSGA to see if it acts as an accelerant or a seed of any sort. And that CSGA certainly does. So even in vitro, we can get alpha-synuclein, the lag phase of alpha-synuclein to go down by about half. If you would have added alpha-synuclein seeds to this, we would have decreased this lag phase all the way down to 0. So we're not as good of a seed or an accelerant as say alpha-synuclein aggregates would be, but this is pretty significant. And then we started looking and we tried to ask the question, are E. coli overexpressed or overrepresented in Parkinson's microbiomes? And there wasn't great evidence for that, but there was good evidence that enterics in general were. And then so Tsujis, my lab, decided to ask, hey, are there other enterics that may have CSGA homologs or the ability to express an amyloid-like protein like CSGA in the gut microbiome? And so he identified several CSGA homologs, not too many. So it was a number, it was 5 or 6 or so that he identified in the first pass. And 4 of these he purified and characterized in vitro. And I'm going to tell you a short little story about 2 of these because they're pretty interesting sides of the coin. So one of these is from a Hafnia species and the other one is from a citrobacter species. And so he purified these CSGA homologs and then characterized their ability to form amyloid in vitro. Both of them form an amyloid, which was nice because he had picked out these homologs on using some amyloid prediction programs. And so both of them form amyloid, which is nice. They form fibers which are virtually indistinguishable from the CSGA fibers from E. coli, but they have very different kinetics. And so the Hafnia species forms amyloid very slowly or relatively slowly, eventually gets into an amyloid. The citrobacter species is a real pain. It forms amyloid very quickly, much faster than E. coli CSGA, which is a fast amyloid former. Citrobacter is speedy quick in getting to that amyloid conformation. So you can measure their lag phases. E. coli lag phase is relatively low, not nearly as slow as Hafnia. And then the citrobacter is extremely fast, much faster than E. coli. Okay, so then we just asked, could both of these or either of them accelerate alpha-synuclein like we had seen before? And so the first one we tried was this Hafnia. And in fact, it does. It's as good or maybe even a little bit better than E. coli CSGA at accelerating the polymerization of alpha-synuclein. Whereas citrobacter was not. There's a little bit of a gap here between these 2 lines. So this is plus the citrobacter and this is just alpha-synuclein. But if we add even more citrobacter protein to this, we actually start to slow down. We actually start to get a decrease of this. And I think Daniel's going to talk a little bit about that, or at least mention something about that too. And so we sort of had this idea that maybe the intrinsic amyloid forming propensity of CSGA proteins governs their ability to accelerate alpha-synuclein aggregation. And so what I mean by that is that maybe these fast amyloid formers don't get into a conformation or don't stay in a conformation long enough to accelerate alpha-synuclein. And the slow amyloid formers, maybe they're more conformationally dynamic and there's some conformation in there that persists a little bit longer and it's able to accelerate alpha-synuclein. And we could actually test that using the CSGA protein directly. And so one of the things that we've known for a long time, my gosh, 2010, so over 10 years, I'm getting old. So one of the things that we've known about the CSGA protein for a long time is that there's actually a few residues in CSGA which actually temper or slow down its amyloid formation. So this is CSGA from E. coli. And so we called these residues gatekeeper residues years ago. And if you come in and mutate these residues, you actually get a CSGA protein that without these gatekeeper residues, which forms amyloid with virtually no lag time, no appreciable lag time. It's really toxic to the cells. So the cells hate this protein when you try and express it. But it definitely has properties which would suggest that this is a faster amyloid former than the wild type version. And this gatekeeper mutant version has no ability to accelerate alpha-synuclein aggregation. So we changed 3 residues here and lost the ability to accelerate alpha-synuclein. And when we looked back, and I say we, I should say Tsujis, because I did not notice this at all. It was totally Tsujis who noticed this. He noticed that this citrobacter protein, which has this really fast amyloid forming ability, it's missing the gatekeeper residues that are conserved in many of the CSGA homologs that we had studied before. And so Tsujis hypothesized that because it was missing those, that's why we got this fast polymerizing phenotype, and that if he reintroduced those, he should be able to slow down citrobacter's aggregation. So he made 3 changes. He introduced 3 aspartic acids, which are these gatekeeper residues, into the citrobacter at the positions that they are in E. coli. And in fact, he made a much slower aggregating, still formed amyloid, but it was a much slower aggregating amyloidogenic protein and eventually got up into the amyloid state. And now if he took this protein and mixed it with alpha-synuclein and asked, could it accelerate alpha-synuclein? Now it does. So this slower aggregating one, citrobacter version, does accelerate alpha-synuclein aggregation. And so this is pretty exciting because now we're really focusing in on these early intermediates as being the things that can accelerate alpha-synuclein aggregation. And so I'll just summarize there so that Daniel has at least 10 minutes to talk. So enteric bacteria can express Curli amyloids and exacerbate alpha-synuclein amyloid aggregation in Parkinson's-like symptoms in that murine model. CSGA accelerates alpha-synuclein amyloid formation in vitro. Several of these CSGA homologs were identified in the genomes of human gut bacteria. Those homologs were purified. 4 of them can assemble into amyloid. 1 of them sort of a pain. And just may not be an amyloid forming protein, which is okay too. Only CSGA variants competent for forming amyloids themselves can accelerate alpha-synuclein. So I skipped over that data, but you have to be just right. You have to form amyloid not too fast, but also you have to maintain the ability to form amyloid at all. If you don't, you can't accelerate alpha-synuclein aggregation. And acceleration of alpha-synuclein aggregation is associated with some sort of transiently formed CSGA species. We have some data with Brandon Rotula's lab, which suggests that that's maybe a dimer species of those small of those CSGA homologs. And then finally, in the older work, CSGC has really potent anti-amyloid activity against CSGA and alpha-synuclein. And we're excited to maybe try and evolve CSGC to be better at inhibiting alpha-synuclein. Remember, it was pretty good, but it wasn't nearly as good as the E. coli version, as inhibiting E. coli version. So, and then with that, I just want to thank all the people who actually did the work. My group is just such an amazing group of people through the years, and then that have been in my lab. I talked a little bit about Sujit's work and Neha's work, and a little bit of Hema's work, who I didn't mention when I got to her slides, so sorry. And then we've got many collaborators across the University of Michigan and outside of the University of Michigan who have just made all of this work possible, and who I'm just forever indebted to. So thank you, and I'd be willing to take any questions if there's time, Wong.
Great talk. Thank you, Matt.
Yeah.
Any questions? If not, I don't see others. Let me start. Okay. So have you started to try to generate bacteria strain that secrete CSGC and put in animals, see if they have any effect?
Yeah. So that's a great question. We have. So we've done that. We're doing that in a couple different ways. So we've got an E. coli strain which is missing the Curli genes, except for a secretion system where we can direct CSGC to the outside. And then the graduate student who's working on this is also trying a couple of lactobacillus in a Bacteroides strain. These are common gut bacteria, and trying to adapt them to also secrete the CSGC protein. And the really interesting thing is it's preliminary at this point, but the E. coli strain, which secretes CSGC, can inhibit biofilm formation of amyloid-dependent biofilm. So I skipped over a lot of this data, but the amyloids are absolutely required for E. coli to form a biofilm. And if you mix in this C-expressing strain, it can prevent that strain from being able to form biofilms because it's preventing amyloid formation by that bacteria. So we think that these strains will work, and we're eager to send them on to Tim Sampson, who's at Emory, because he's going to colonize them into those synuclein overexpressing mice and see what happens. It'd be really great if we could keep those bacteria in the gut, prevent alpha-synuclein possibly from forming amyloid in the gut, and then if that translated to the disease, that would really be a nice connection of this of these two things, I think.
So I see someone from Samsung Galaxy. I don't see his name, but can you just talk?
Christian Grisinger, go ahead.
Sorry, I'm connected by a phone. I could not manage via computer. I'm really sorry. I have a question. So, I mean, you show this co-aggregation or aggregation enhancement of Curli of alpha-synuclein, but how do they meet in the mouse? I mean, the bacteria in the gut and alpha-synuclein is in the neurons. And
yeah.
So, I mean, do you think there's a spreading through the gut wall and so on involved, or how is that occurring?
Yeah, it's a great question, and I don't have a great answer for it. But several things happen, during the dysbiosis that precedes, the amyloid formation of alpha-synuclein in the brain. And one of those is sort of the gut barrier becomes compromised. And there is exchange, between that vagus nerve and the gut of proteins. Probably not, it doesn't take the whole bacteria. The whole bacteria is not, clearly that would lead to other problems. It's clearly not the whole bacteria which would be doing this. But you could definitely have an exchange of proteins. We have not shown that directly. We tried a little bit, and we're still working on it. We would love to have that sort of smoking gun in the gut. We don't have that yet.
But I mean, could there be also alternative mechanisms like, for example, biofilm forming bacteria, maybe induce oxidative stress?
Yes, induce inflammation and an inflammatory response. Yes, you're spot on, and absolutely. We haven't discounted that. You might have seen Curli themselves are pretty potent inducers of certain parts of the inflammatory response. And so absolutely positively, it could be through, or at least in part due to that inflammation that's caused by the bacteria. Interestingly enough, though, LPS and bacteria themselves, these gram-negative bacteria, are great inducers of the inflammatory response. And so it really would be a Curli-specific or amyloid-specific inflammatory response in that case, too.
So if you take LPS, you cannot induce alpha-synuclein aggregation.
You don't get the same response that we got with the Curli. Yeah, absolutely.
Okay,
yep.
Thanks a lot.
There is a question from Olga Gursky in the chat box.
Oh, let's see.
I can read it for you. Do you think that CSGA amyloid acts as a template that helps nucleate alpha-synuclein amyloid formation and growth?
Yeah, that's what we thought at first. We don't think that is necessarily the case anymore. Number one, we don't see the interaction between alpha-synuclein and CSGA. We haven't been able to show yet that interaction, if CSGA was forming an amyloid-like template and then alpha-synuclein was sort of building on top of it, we would, easily be able to, sort of visualize, or we thought we would easily be able to visualize sort of those heterogeneous fibers that contain both proteins. We don't yet have any evidence of that. We see alpha-synuclein apparently getting into an amyloid-like form more quickly and assembling into fibers more quickly. So we can do this by single molecule fluorescence studies. But we haven't yet seen evidence that like a CSGA is stuck on the end of that or on one part of that, forming a heterogeneous fiber.
I have a question, Matt. Good morning.
Hi, Raj.
So
Good to see you.
good to see you too. Where you stand on the whole viral infection with Alzheimer's, microbial urinary tract infection in Parkinson's,
As a microbiologist, it gets me excited, right? It combines, two of my loves in some way, and two of the things that I've been interested in for my whole career. I agree with the skepticism that I can hear in your voice, or the, where's the actual evidence? I think the more that we learn about the role that these microbes play, I mean, just think about it, just for a second, like all of that genetic information, all of that, all of those proteins, all of that business that the microbiome is doing, it is certainly influencing in many ways that we have only begun to understand what's happening to our, what we would call our normal physiology, to our proteins, to what our cells are doing. And so I think there are links. I think there are pretty clear links, especially in Parkinson's disease. The A beta, I think there are clear indications that there probably are links there too. And so I don't think we should discount what that microbiome is doing and its influences on the sort of normal physiology of our cells and proteins.
No, as an amyloid, sorry, just follow up. As an amyloid guy, I like it. I think it's, as you said, it's very exciting. But the hypothesis like amyloid or A beta is antimicrobial, etc. So it's just still no.
There's not great evidence, but right. But it's even in that case, with those stories, amyloid, as we all know, is cytotoxic. And, our immune system is a good example of this. Of course, it's cytotoxic. Well, it also causes cytotoxicity to our own cells. And so our immune system is a great example of our bodies, coming up with defense mechanisms for the microbial world that don't necessarily only target the microbial world. And so is it possible that something like these amylogenic proteins that are secreted by cells and on the cell surface, that they could be in some way targeted as cytotoxins to bacterial cells? And then, of course, there's collateral damage with that and could affect human cells. I think the possibilities there, and I find it a really intriguing line of thought. Again, in that case, I don't think there's a real smoking gun that would say, it's really hard to go back in evolutionary history and say, okay, this is why A beta is here, or why we continue to have A beta as part of our genomes.
Thank you. Great talk.
Yeah, thanks. And should I introduce Daniel?
Hold on. Ina has a question.
Okay.
Hi.
Hi.
Matt, can you hear me?
I can hear you great.
Yes. Thanks for the great story. I have a question regarding the mechanism you think that works when CSGC acts as an anti-amyloidogenic, because usually when you want to keep a protein unfolded, you would have a complex formation, and also because you said CSGA, as soon as it comes out, it will quickly again form polymers. So, and with such low concentrations of CSGC, it's like catalytic. It's just working it, or is it moving it into some conformation which is very different, cannot polymerize? What's your thought?
It's a great question, Bina, and I don't have a great, really clear answer, but we certainly have some speculation. And so our favorite hypothesis at the beginning was that CSGC was capping some sort of, some sort of oligomeric state of CSGA. So CSGA would be getting into a fiber, and then CSGC would just cap that. And if you look at CSGC, the structure really led us there, because CSGC, for all the world, looks a lot like CSGA. It looks like a cross beta strand protein, but the difference is that CSGC is a non-aggregating protein itself. It is the most well-behaved protein we've ever had in the lab. It stays in that dimer state, or at least monomer or dimer state, for as long as you want to keep it. It never aggregates into amyloid. And so, and then a prerequisite of that capping model would be that should form a relatively stable species. Maybe you do have 400 CSGAs and then you've got a cap. That does not happen. We have never seen any evidence of that ourselves. And so if you take CSGA and mix it with CSGC, CSGA stays as a monodispersed and intrinsically disordered protein, and it will stay that way for weeks along with CSGC. When we try and measure the CSGC-CSGA interaction, it is not very strong. It is incredibly weak and very dynamic. So we think the on-off rate of CSGCs and CSGAs, that interaction is really transient and relatively weak. And so our best hypothesis, and it's just speculation, is that there is a particular species that in a conformationally dynamic pool of CSGAs, where one CSGA may get into a conformation that CSGC is able to recognize, and CSGC transiently interacts with that CSGA, bangs it back into the disordered conformation, or whatever conformation that is, and then lets go and is now free to maybe interact with the next CSGA, which comes to that crest or comes to that conformation. And we think that must be how CSGC is working. Of course, it's very hard to get at these transient weak interactions, and it's also hard to get at these conformationally dynamic species. We're trying to pick one of them out. It's a difficult question. Graduate student in the lab right now is toiling away at that very question, though. He would love to know what that mechanism is. And we've gotten a little ways, by doing some mutagenesis studies that I didn't talk about, but yeah, we would love to know more about that.
Have you ever tried to measure binding kinetics of the two?
Yeah, you can. So CSGC actually will interact with preformed CSGA fibers quite well. So it'll interact with those with measurable kinetics. That inactivates CSGC. Then CSGC cannot act as a chaperone. It's stuck to those fibers. We have not even been able to measure the kinetics or the KD of the interaction between CSGC and intrinsically disordered protein. And I think there we just have the problem of it's such a transient interaction and happening in a population. We've only tested in population experiments, and when we try and do that, we just don't see it.
Okay, Matt. Thanks a
Yeah.
lot for the good talk.
Yeah.
Excellent. Now you can introduce Daniel here.
Yeah, so I'll introduce Daniel. So Daniel, I've known Daniel for a long time, and I'm sure that this whole group does. He has been a prolific figure in the field. And so Daniel, he's a professor of nanobiotechnology at the Interdisciplinary Nanoscience Center, or iNano, at Aarhus University. He did his PhD at Cambridge, and then he took 2 years. So graduate students that might be on the line, there's lots of different routes to in scientific careers. And Daniel worked as a chemist for a couple of years at Novozymes. After he finished his PhD, he looked at the impact of surfactants on protein stability and structure. And this really kind of got him interested, I think, career-wise, in the role that protein fold, or how proteins fold and how they're interacting with their environment, and what happens when things go wrong. He did a postdoc at the University of Lund with Mikhail Alberg, and then he joined Aalborg University as a professor. And then in, I think, 2007, although I forgot to write that down, but 2006, 2007, he moved to Aarhus. Yeah. And if you go to Daniel's website and try and scroll through his CV, you'll get like scrolling thumb or whatever it would be called, because he's got so many papers. It's in the hundreds of papers. And if you have not had the chance to just sit down and enjoy a conversation with Daniel, please do so when we finally get back to real meetings. And he is just one of the most warm and funny and welcoming individuals on the planet. So I'm looking forward to your talk, Daniel, and well, thanks.
Well, thank you very much, Matt.
Matt, you have to stop sharing.
Oh, yeah.
Great. And so let me just get this going. Right. Thank you so much, Matt. I blush at your kind words. In fact, my computer tended to freeze over when you were particularly, panegyric in your words, and I think it's really, really kind of you. And you have a great future as a PR manager as well, as Wang mentioned. And in fact, I can return the accolade with complete honesty, because it was Matt who got me into this field, as you will see. And Matt's slide about CSGA is basically how I start all my talks. And I have an interest in the bad side of protein folding and aggregation, but functional amyloid represents sort of really the nice aspect, the useful aspect. And today I'll basically continue this wonderful talk that Matt gave and the ensuing discussion, which clearly centers more and more also on a molecular understanding of what's going on. So in this whole process of making a functional amyloid, we'll be looking at how you start the process, how you elongate them, and possibly also how they break into pieces. But as I mentioned, sort of Matt's work is seminal in this. It was reading this while preparing for a lecture in 2003, I think, that I basically started working with functional amyloid. And as you know, Matt showed 2 decades ago that the Curli operon is based on formation of useful amyloid under very controlled circumstances. You have this wonderful orchestra of proteins collaborating under controlled circumstances to make amyloid. On their own, they just form jumbled fibrils, but together they can really control it in space and time. And this is also in the form of strengthening the biofilm, as we will see. There was a lot of discussion that Matt raised about also the role of amyloid from the microbiome. And just as a small snippet of that, I should briefly mention that we've been working to also see what amyloid forming proteins there are in the microbiome of rats. And we've recently published this, so I won't go into details. We identify proteins by virtue of the fact that amyloids are so tough that they only solubilize in high concentrations of formic acid. And in this way, sifting through the data there, we were actually able to identify a number of proteins. Our own limited capacity prevented us from going into depth with all of them, but we did identify some that were also exported and found that they are actually, well, they live up to the hype in a certain way, in the sense that they are certainly able to form aggregates under physiological conditions in a very efficient manner. So I think there is a lot to be said for going hunting in the microbiome for functional amyloids. I think Matt made a very strong case for CSGA and CSGA homologs, but there may also be others if you look. But anyway, what I will talk about more today is essentially how you make amyloid in vitro or at the molecular level. And we use Matt's Curli operon and also an operon that I discovered with my colleagues at Aalborg more than a decade ago, where you have a number of proteins collaborating to form amyloid through this protein FAPC. And the whole process has been started also with Steve Matthews, who's a very important member of this from a structural perspective. There are certain channels that transport our FAPC across the membrane, and FAPC also aggregates in a very prolific fashion. And the upshot of that is that when you make biofilm that contains this kind of amyloid, you simply strengthen it. So you can press down with a cantilever and a small bead to show that the presence of, or the overexpression of amyloid in this biofilm makes it something like 20 times as stiff from in terms of the so-called Young's modulus. So as Matt says, it's a vital component in the formation of strong mechanically resistant biofilm. But know that, so we have a system that really works well. And how does it work well? Kind of in the Feynman approach, I can't understand something unless I can take it apart and hopefully build it again. We've been collaborating with Thomas Knowles in Cambridge to use his tools to understand the molecular mechanisms of fibrillation of this functional amyloid. And Thomas, and together with Sam Cohen some years ago, developed a program, AmyloFit, which allows you to find out which of these types of steps can contribute to amyloid formation. Obviously, you need to have nucleation to start the whole process. Then you extend the nucleus to actually grow your fibril. But then you can have so-called secondary processes where the fibrils either fragment or allow growth along the sides, they catalyze growth laterally, so that you get this exponential increase. And looking at the kinetics, how proteins aggregate at increasing concentrations of proteins, you can work out which of these steps are going to be most important, most prominent. Is it mainly driven by primary processes, just a slow and steady, or does it involve these more kind of explosive feedbacks? And the long and short of it is that the functional amyloids, they are basically on the straight and narrow. They are happy enough just to do primary nucleation followed by elongation. There isn't even any much significant amounts of dissociation. We don't have very much of fragmentation or other secondary nucleation. So they are largely focused on extending fibrils once they've been formed. And you can show this by showing that if you try to include secondary nucleation in your kinetic normalized thioflavin T curves, they fit really badly. Whereas the ones that just involve this fast track fibrillation without any sort of digression into secondary nucleation or so on, they fit very well. And this isn't a simple party observation. It truly appears to distinguish functional amyloid from pathological amyloids such as A-beta or alpha-synuclein in our hands too, where you see this much more explosive increase, indicating that you have this feedback that basically allows it to go into an aggregation frenzy. And this whole distinction between primary nucleation and secondary nucleation appears to be a defining feature of whether amyloid is good or bad. So whereas there is neither good nor bad, but thinking makes it so. In this case, it's primary versus secondary nucleation that are processes that appear to provide the answer. So, for example, our functional amyloids are in the lower part of this graph, indicating that the primary nucleation, the X value, is higher than the secondary nucleation, whereas bad guys like A-beta and alpha-synuclein, or for that matter, sickle cell hemoglobin, are in the wrong part of the graph there. And so they've been tuned or biologically programmed to do this. And a clue as to how this occurs is to be found in their sequences. Matt has already alluded very nicely to it. Both the FAPC and CSGA have multiple imperfect repeats. In fact, CSGA is a very parsimonious repeater, you might say. It basically just consists of repeats, whereas FAPC is a little more splurge-oriented and has these long linkers of variable length. In fact, the number of repeats in FAP vary across species, ranging from 3 in where we found them in Pseudomonas to as much as 14 in this exotic marine organism, Vibrio brasiliensis, which has a staggering 14 or more of these repeats, indicating that it's really, really going for amyloid formation. I can't go into details now, but amyloid is also a kind of sponge that retains nutrients in the region of the biofilm. So it makes sense that marine organisms, if they try to form communities under those conditions, they really have to hang together and keep all the nutrients in the vicinity. But we are molecular biophysicists if we can get away with it. And that's why we try to ask molecular questions and ask, for example, what happens if we remove one or more of these repeats in FAPC? And the answer is actually relatively straightforward in that you increase the tendency to fragment. So you increase the tendency to go to secondary nucleation. Here we have a number of repeats where we've removed either repeat 1, 2, or 3, or combinations of them. The third one, or the last one, actually has all repeats removed, and it actually aggregates still. And there is a simple bioinformatic reason for that, as I'll get back to at the end. But it does so in a much more chaotic way, and the fibrils are basically pathetically weak, as you will see. But the interesting part is that we can use our magical amylofit program to identify to what extent fragmentation contributes. There's a so-called critical fraction. That's the concentration of the population where fragmentation becomes more productive in making fibrils than the original nucleation and elongation. And we can define this and show that for wild type and just a very modest removal of one repeat, you stay in the nucleation-dominated regime much more than for the other mutants where you remove more repeats. So they tend to fragment more easily as you remove more and more of these repeats. They become basically, in a sense, they of course, that also leads to more secondary nucleation, so that they could in principle also aggregate faster. But the fibrils clearly do not become as long. And that's also an issue because we want long fibrils to allow our box to entangle and make biofilm. We can show this because we are basically, I'm a protein folder by training, so it's in my blood, metaphorically at least, using denaturants. And we've shown that using formic acid to basically quantify stability of fibrils, we can show that the midpoint of denaturation, which is a respectable 66% for wild type, plummets to basically negligible as soon as when you remove all repeats, and goes down before then as well. So the repeats both make aggregation more orderly and lead to more stable products. You can actually quantify this even more. We can look at how formic acid destabilizes also globular proteins. So the sensitivity to formic acid concentrations, the so-called M value, tells you something about how many residues are involved in being buried when you fold or form aggregates. And this scales rather nicely with the number of residues in the protein. So in other words, the higher the M value, the more residues are buried to form amyloid. So you can, in a sense, estimate the extent of residue burial and thus amyloid formation by looking at the sensitivity to formic acid, so the slope of the free energy of unfolding, as it were. And we can deduce that from our various repeat mutants. And you have a very busy slide here, but that tells you that the not only does the midpoint of denaturation decrease as you remove more and more repeats, but the number of repeats involved, the number of residues involved in amyloid formation also decreases. as you can see here, though, I mean, not by an astounding amount, but clearly it becomes, in a sense, less involved in the process, except, funnily enough, at the very end of the list, where you start to remove more than a critical number, you get what appears to be a strange turnaround. I'll get back to this later to explain basically that you get another type of fibril. So it's nicely packed, but it's extremely unstable. So you basically induce polymorphisms or alternative types of aggregation when you mess around with your aggregate. So these repeats are very important, not just for driving an orderly emphasis on just nucleation and elongation. And if you have few of them, they tend to fragment more and more, but they also lead to, they stabilize the ensuing fibrils and make sure that they can stick around and play its role in the biofilm. So the repeats, given that they are so interesting, it would be nice to know what they look like. And Matt has already nicely shown the sort of at this stage relatively hypothetical structure of CSGA. The wonderful thing, as I said before, about working with functional amyloid is that you have biological information. It's not just a cop-out, a defeatist, misfolded state. It's actually something that biology has optimized. So we have lots of sequences we can get information from. So we can do bioinformatic analysis to compare different sequences of, say, CSGA and find out which residues basically accompany each other through evolutionary history. This essentially provides structural constraints because it tells you that residue, for example, in this case, you can see residue 20 odd, something is in contact with residue 40 odd here, as predicted by two different programs. And that allows us to feed into a prediction mechanism or machine where you combine this sort of co-evolutionary conservation with a normal force field to predict structures of proteins. And so you end up with a structure of CSGA that is essentially a rather beautiful beta helix, where each repeat corresponds to one beta hairpin. Start here with the so-called N22 sequence, which is not part of the final structure of fibrils. And then you have the first repeat in red, yellow, purple, green, and then blue. So in that sense, each repeat presents a rung that just can stack on top of the other. The slightly frustrating aspect at that stage was that we were unable energetically to distinguish between left or right-handed beta helices. So we would need more experimental information about this. But the fact that individual repeats were able to form beta hairpins makes perfect sense. Again, harking back to Matt's work in the noughties, where Xuan Wang from his group did some beautiful work showing that individual peptides were able to fibrillate, at least those in the termini 5R1 and also to some extent R3. So it makes sense that they are sort of semi-autonomous folding units. We've subsequently, or rather Madhu, my postdoc, has shown, working with Christiane Ritter in Braunschweig, that you can do HDX exchange of CSGA and identify which parts are protected. And they fit beautifully with this structure, showing that the beta hairpin that we predict also is the part that's most protected against hydrogen deuterium exchange. So that nicely vindicates it. Madhu has also done more real solid state NMR work on CSGA fibril structures using some clever isotope labeling, and we are hoping to get that out relatively soon. It's based on his work in Christiane's lab and then continue to a small extent in my lab. And the interesting thing is that you need to combine this NMR, the spectral information, with MD and co-evolutionary constraints, but then you can pretty firmly establish that it is a right-handed beta helix, as one would expect. That's basically what nature prefers. So we have a pretty good stab at a structure for our functional amyloid, which shows this beta helix that obviously would imagine to be stacked on top of each other. Rather maddeningly, we still haven't got clear evidence as to whether it's head to tail or head to head, but head to tail would be the simplest option, I suppose. FAP-C, based on the same approach, also seems to form these beta hairpins, and the linkers then, they are sort of just hanging around, it seems, making a bit of a sort of frizzy surrounding to it. But they have other roles to play as well. But let's go from structure to mechanism. In other words, understanding how the aggregation occurs, and not least how it can be influenced by other species or by other components such as chaperones. And it's clear that CSGA is subject to chaperone regulation through CSGC. Again, using the amyloid approach, you can tease out what parts of the aggregation process are affected by chaperones. And the Knowles group has shown some time ago now that different chaperones, such as these mentioned here, have different effects on the same protein or peptide, A-beta-42. What you can do is simply look at the THT time curves, and then you can also almost visually see that if you have just inhibition of the primary nucleation, you just displace your time curves like this. And if you have other types, you have other features there. So what we've done is to take our functional amyloid, because we were curious to see whether our functional amyloids were subject to the same rules of engagement, as it were, as pathological amyloid. And we looked at TTR, which Matt has collaborated with Joel Buxbaum at Scripps to show is a pretty potent inhibitor of aggregation, particularly in the monomeric state. There's also other proteins like S189, which is a bit of a funny character here because it actually is toxic on its own, but it can, to some extent, mitigate its own and others' effects by going together. So too much of a good or bad thing can be a good thing here. Then there is the BRICOS or the BRI2 domain that Janne Johansson and Henrik Beverstål at Karolinska in Stockholm have also worked a lot with. There's a bona fide chaperone, DNA JB6 from Cecilia Marlton in Lund, and CSGC, as Matt mentioned. And what we've done, and we're going to, don't worry, I'm not going to go into details with this slide when we get to it. It's a rather busy slide, but we've simply taken all these chaperones and subjected CSJ and FAP-C to aggregation in their presence. And there is an effect, and it varies a lot between chaperones. And to make sense of all this, we've tried to see whether variation in either the rate of nucleation, secondary nucleation, or elongation is what best describes the effect of the chaperone. In other words, does the chaperone decrease the nucleation, the secondary nucleation, or the elongation of the whole process? And rather nicely, you can see that no matter which chaperone, and they are all very different, you compare, you get a clear preference for a model in which it's the nucleation that's targeted. So in other words, it's the very early stage of aggregation that these chaperones are best at inhibiting. And I mean, that, in a sense, it's a bit of a no-brainer, because clearly if you want to have an impact, you want to nip the evil in its bud. So basically stopping the aggregation from proceeding further. This also implies that once you have seeds of your fibrils, in other words, the nuclei have been formed, you can't make much headway with chaperones. That's actually the case with, for example, these chaperones, DNA JB6 and monomeric transthyretin. You can see the red curves are basically the same as the black curves with and without chaperones. So they seem to be powerless once there are seeds present. But that's not always the case with our other functional amyloid, FAP-C. They do have an effect even when there are seeds present, indicating that if they don't go for this one, the chaperones, they can also target, say, the elongation. But returning to the function of the nucleation step, well, you can imagine that you could either target the monomer, and that would make sense if you have to have stoichiometric amounts of your chaperone present, as is the case for these guys here. Whereas if you are you have CSGC or this chaperone DNA JB6, which are very substoichiometric. Matt's numbers were even lower. We find that we may not have a super good CSGC as his size, but it's still substoichiometric. Breed 2 is sort of in between. But as Matt also mentioned, CSGC, it keeps CSGA unfolded, so it doesn't seem to induce some kind of parked beta sheet rich state. So the question is whether it recognizes this transient protofibrillar structure that the nucleus presumably must form as soon as it starts to form, and then binds to it and kicks it off once it's been able to prevent it from getting any further. To analyze this, we actually tried to measure the affinity of our chaperones for monomers using Biocore immobilization. And I'm intrigued that about Matt mentioning the very dynamic nature of the interaction. In our hands, it's true that the binding was not super strong for many of them, but we could actually get some relatively low KDs for our CSGAs and even more, as you can see, for our FABCs in this way. It may, of course, be that immobilization induces a transient structure which increases the affinity. We can't rule that out. But by and large, we were able to see, and in this rather incoherent graph, that there was a trend that the lower the affinity, as you go further up the x-axis, the lower the impact of the chaperone. In other words, for a chaperone to be effective per molar chaperone, it needed to have a relatively good binding to the monomer. Which part of the monomer then? Well, you can do peptide array to display different parts of your protein. And interestingly, we find that both the CSGA and FABC, the chaperones seem to like the same parts of the protein. They bind to some parts and not to others. This is a very coarse peptide array. We don't really do a very nice staggering array of the different peptides. So it's we can't deduce too much, but there is an indication that they all recognize more or less the same parts in the unfocused. Sorry? Okay. And another thing is that the chaperones, they actually work in vivo as well. So we can measure how well they inhibit biofilm formation using simple PEG assays. And we can show that as you increase the affinity for our functional amyloid, you also get significant decreases in the amount of biofilm being formed. But that puts us in a bit of a spot because we know that DNAJ B6 cannot, at least in vitro, inhibit seeded growth. So once CSGA starts to form, you would expect that DNAJ can't be active here. So the question is where DNAJ actually works in vivo when it does this. And we can't really say this for sure at the moment. What we are hoping to do at some stage is to use a new stealth weapon developed basically from, well, originally from the Linköping group of oligothiophines, and also now more made commercial in a company, Ebba Biotech, by Agneta Richter-Dalfors and Ferdinand Chong, who have developed these nice biomarkers which can trace bacterial amyloid growth, and they have a very a long wavelength fluorescence, so you can distinguish it from other parts. And they've shown that you can definitely detect Curli in this way, also from individual Curli as the E. coli cells expand. You can even do neat stuff such as looking at the when the Curli starts to form, where it forms in the biofilm, how the various parts of the biofilm are columnar or are filled with amyloid or not, and of course the kinetics of amyloid formation over time. So we are hoping that this might be a way to analyze this in more detail. But I know that I'm running out of time, but I'd just very briefly like to mention a final twist on the analysis of how our aggregates form in vivo, namely going back to the ultimate way of looking at protein folding and unfolding, namely chemical denaturants. So chemical denaturants are like stabilizers in reverse. They like to bind to the protein. And that means that basically they encourage a very expanded state of the protein. The less compact, the more expanded, the more denaturant can bind. And so this M value that I briefly alluded to, which explains the sensitivity of a given process to denaturant, tells you something about the level of compaction. So the greater the destabilization by denaturant, that implies a greater binding of denaturant to protein and a greater increase in surface area upon unfolding. So they can basically tell you about changes in compaction, both during unfolding, folding, during kinetics, and why not during aggregation either. So denaturants have been used to probe amyloid stability in the group of the late Chris Dobson, as you can see earlier as a decade ago. We've subjected aggregation of FAP-C and CSGA to the kinetics of aggregation to different increases of denaturant. So guanidine is far too efficient, so it doesn't really work in our hands, but urea is a nice one. We get a decrease in THT fluorescence, which need not concern us too much. It's simply this sort of weakened binding of THT, because all the protein, as you can see, largely aggregates even at very high urea concentrations, and we get fibrils. What we see, though, is that if we look at which part of the aggregation, nucleation or elongation is affected, they are both pretty much equally sensitive to denaturants. And that's a pretty important observation, because it implies that both the nucleation, which is the primary part here, and the elongation, which is the growth, they have equal levels of folding. What it means in practice, I guess, is it's still up for interpretation, because it implies that the nucleation that involves a certain amount of burial of surface area, possibly by a number of molecules coming together, or simply by the protein itself folding. We know the nucleus is very small. Matt mentioned a possible dimer, so it really is very small there before it actually forms a fibril. But clearly there is a kind of they are closely linked in terms of how they fold in this process. So elongation is in a sense an extension of nucleation. Important, we can also show that urea doesn't change the actual aggregation mechanism of whether you do it at 0 molar or at 4 molar. You can fit it to the same model here. So it's not as if we are for a normal protein that we are impacting this whole process. Let's see. What we can also ask, of course, is which parts of the process are being affected most. And again, here we can turn to amylofit and see which parameter variation best explains what we see. And funnily enough here, unlike before where it was mainly nucleation that was affected by chaperones, with urea, it's the elongation is slightly more is better than at fitting the process than nucleation. So it seems that urea is placed just as much, if not a greater impact on the subsequent elongation step as the nucleation, again emphasizing that elongation is very much part of the whole sort of folding process here. We can also confirm this using a seeding process where we just focus on the elongation and show that you get a sensitivity of your elongation to urea. And the final twist here, before I finish, is that curiously, you actually see that when you start to mess around with your, in this case, your FAP-C by removing repeats, you get larger M values. So the sensitivity to denaturation increases, indicating somehow that messing with the FAP-C by removing the repeats leads to fibrils which bury more surface area when they form fibrils. We can actually confirm this using small angle X-ray structure scattering, which allows us to model both how the initial state looks like and what the fibrillar state looks like. And the initial state actually cannot be modeled as a monomer because it starts to come together even at the initial stage, basically as soon as we've taken it off the purification column. But they are not fibrils at this stage. Interestingly enough, the early stage of the deletion mutant is more compact than the wild type, but they are still basically very expanded. The final fibril is actually larger though for our deletion mutant. So it's actually there's a bigger increase in compaction because the fibrils are larger. And what does this mean? Well, apart from the fact that even though they are compact, they're only half as compact as at best as globular proteins. So they're not super well packed. And it implies that whereas the wild type is able to form strong fibrils, even though they only involve part of the sequence. Once you have truncations, that means that you basically have to aggregate what you can aggregate. You form larger fibrils, but they are not, which are buried, but they are not buried in an efficient stabilizing manner. Obviously, that's something that would be nice to look at more clearly by, say, cryo-EM. We are also looking at this together with Tanmay Bharat at Oxford University and Jan Böning. But it implies that even though we have, when we look at the aggregation propensity of, in this case, FABC, and ignore, of course, the signal sequence, we have our repeats here, but the linker regions are actually also relatively aggregation prone, but they don't get their time in the sun unless we remove everything else and then allow the what's left to aggregate. So implying that they have a weak aggregation propensity on their own, but if you let the real repeats aggregate, they will basically run with it and then leave the others hanging around as well. So to conclude this relatively long spiel, I can say that I've mentioned that our amyloid is formed in an extremely well-coordinated manner, and it's a straight and narrow fast track involving primary nucleation rather than secondary processes, unless you start to mess with the repeats. And the repeats are basic folding units which form beta hairpins, and they, in a sense, promote nucleation and elongation as the main processes in fibril formation. And you can mess with them by removing them, leading to altered fibrils, so that you increase the polymorphism, you might say, or the access to different types of fibrils in this way. So you should basically stick to nature's recipes rather than mess with them if you want to maintain your good fibrils, at least from a bacterial perspective. I've mentioned people along the way. Here are also some of the other people involved and those who support this. So thanks very much to my support, and also obviously to you for being present. I'm very happy to answer questions. Thank you.
Daniel, maybe I'll ask a question if nobody else is speaking up. Really stunning. That was amazing. In your Biacore experiments or SPR experiments where you were measuring the the interactions, I think we're on the right path. My lab might be, because we've done some of those too. So it's good to see that you have also, and we saw that sort of same result if we immobilized what we think were fibers, most likely, onto the Biacore trip, and then flowed over CSGC. That's where we saw a nice interaction, or at least a measurable interaction. Did you try and do the reverse experiment? It's the reverse experiment that we could never get to work, and I'm just wondering if you had any better luck.
You mean where you immobilize the chaperone and then measure binding?
Exactly, and hopefully flow, monodispersed, or as close to monodispersed. I know it doesn't, the problem with it is it doesn't stay that way very long. But if you do that reverse experiment, that's the one that we've struggled with, or at least to see an interaction.
Unfortunately, we haven't done that, and in fact, I also think that we should do the fibril analysis as well, even though it's maybe more important to look at the initial stage. But unfortunately, we had the very practical question that we were dependent on generous donations of our chaperones, and we tended to run out of them rather quickly. So there's a very mundane and not very useful answer to that. But I agree that it's something that should be done. I think that immobilizing the so-called monomer may be our best bet, because I don't really think that it's a pure monomer, but at least it prevents it from aggregating much further this way. So we get an approximation.
I like that. Monomer. Yep. Yeah. I like that. Yeah, that was really stunning. And I see there's other questions, so I'll be quiet for sure.
No, thanks a lot. Thanks a lot, man. I think Astrid has a question.
Yes, well, thank you very much for a very interesting lecture. So I have a question. which is about the role of membranes in all of this. I mean, you are in the real life, so to speak, everything is happening in the presence of membranes. And do you find any differences, and would it depend on, say, head group charges and things like that?
That's a great point. So we've tried to look at having vesicles present. It's just a bit difficult to work with. We didn't get any real clear answers there. What we do find is, in any case, it's probably not, it's just as much the LPS that was discussed in the wake of Matt's talk. The LPS is the outer layer, at least in E. coli, which is what's close to our Curli, and we can definitely see an impact of LPS on that. That strongly accelerates aggregation of our CSJ and FAP-C. It's a good question. Whether it's to what extent it is important biologically, because I would assume that it's able to diffuse out CSGA and FAP-C and then find the ends of the
and then find them again and use them somehow. Yes, yeah, very interesting. The whole story here. I'm sure you will find many interesting things as you come further.
Yes, thank you. I hope so. I think also these oligothiopine probes of amyloid formation can be very useful.
They will be very good. Yes, that's true.
A great Swedish invention.
Yes.
Daniel, when you measured the binding affinity, you mentioned about the monomer being is the one you're looking at. So what happens if you have oligomers? That's one. What happens? Is it also depending on the concentration of the protein? Does it depend on the concentration?
Sorry, oligomers of our functional amyloid, or
yeah,
so yeah, that's a great question, and as Matt would say, I'm afraid the answer is not as good. His answers were good, I would say, but the thing is that it's very difficult for us to actually get oligomers, at least quantitatively. We see oligomers, but they don't seem to be very well defined. We can pin them down by making oligomers in the presence of inhibitors. I didn't talk about this, but we've looked at how the usual polyphenol inhibitors like EDCG and PTG, they stabilize, at least transiently, oligomers of our FAP-C and CSGA, which can then be studied, and they seem to have like a structured core and then a fluffy area. But I'm afraid that the relevance in this to your question would be limited, because they sort of are natural or artificial in that sense. So I'm afraid in practice we are only left with having these wannabe monomer species versus the fibrils as relatively stable species. Anything that we try to get our grubby hands on and we think is in between is likely to be on the way to forming fibrils.
Thank you. Rakesh, go ahead.
Yeah, great talk, Daniel.
Thanks.
So
all right,
my question is, it's amazing that you get the different fibrils or the polymorphs, but is one more dominant? Are there some conditions to keep them together, coexist? Anything in that regard we can learn from?
Yeah, well, I think the problem is that we do not. Perhaps I was being a little bit over enthusiastic when I said polymorphic. It's polymorphic in the sense that mutants certainly seem to make different fibrils than the wild type, at least judged from our analysis of their stability towards formic acid and so on. Within a given species, they also seem to be a rather mixed bag, but I think it's the issue there is more that they tend to bundle together a lot, at least in our hands. We don't get nice, clean, separated fibrils, and so that's a real headache for the cryo-EM experts, because you obviously need to have separated fibrils, and that there's also the prerequisite for saying something intelligent about polymorphism. So what we are trying to do is to use different detergents to keep them separated. Detergents which, of course, do not encourage fibrillation on their own, so they have to be uncharged, but they seem to be able to do that. And Perhaps if you ask me that question in half a year's time, I might be able to say some more about this, because then we can also directly compare fibrils formed by the different species or the different mutants. Great question.
Very great talk. I have a question actually for both of you, like on this functional amyloid. So looking at these great results, it actually took me back, like for a few months, I was working on the East of 35, which is also forms like kind of functional amyloids. So my question for you and Matt is that I was wondering, like, have you ever tried to do, maybe in literature, which I'm not familiar of, have you ever tried to look at like how the temperature and the stress can actually shift the amyloid core for this kind of protein, for example, CSG and PAP? Because like I know that people have done kind of like digestion experiment where they can form the amyloid and they can just incubate with clip B or something, and they can actually estimate how the amyloid core is actually shifting from one region to another region and how they're actually connected to the function of this protein. So I was wondering, like in your case, for example, in your PAP case, you said that when you deleted some of the repeats, then you can get different kind of polymorphism. And when we are talking about polymorphism, we are not talking about only the cross beta structure or anything. We are also talking about the frame shift between the amyloid core, like the region that forms the amyloid. So can you just provide some insight to us on that?
I'd certainly love to. It's something that I've been wanting to do. I'm sure Matt probably has a more intelligent answer than me, but unfortunately, we're challenged by the fact that like a protease profiling of our fibril would be great. We haven't, but the problem is that once the fibrils have been formed, they tend to not go into solution unless we add formic acid. That's not that doesn't, in a sense, prevent us from actually measuring what's left. We just haven't gotten around to it. But I think proteolysis profiling would be the best way to start looking at the polymorphism. We've done it for alpha-synuclein fairly recently, and of course Ronald Melke is an expert at doing this for his alpha-synuclein variants. So I think that also in addressing Rakis's question, this is something that we should do. We just haven't done it, but it's not insurmountable. We just haven't put the resources aside for it. Matt,
how about the temperature? Like if you make the fibers in 37 degree and 4 degrees centigrade for Fab, do you expect like some kind of frame shift in the fiber region?
That's a good question. We haven't looked at it, but I think that seeing to what extent temperature would affect not just the rate and the outcome, but actually the structure of the fibrils, that would be very interesting. I wouldn't put money on it. I would expect it to be certainly a wild type to be pretty stable and consistent, but I'm willing to be shown otherwise. But I would just imagine that Curli is formed. Well, it's largely formed at low temperatures, at least at 28 degrees and below. If you want to get a good amount of Curli, I don't know whether that's because there are alternative ways of stabilizing the biofilm at higher temperatures. So, as you can hear, it's speculation on my part. So, Matt might. What do you say?
Yeah, really quickly, we did some of those way back when, and so did Bill Kay's lab pretty early on, the proteolysis experiments, and you can add to CSGA once the fiber forms. That core consists of those repeating units. Proteolysis will extended incubation with the proteases doesn't affect those fiber cores. It'll clip off that N22 that Daniel very quickly mentioned. It'll also clip off just a little bit at the end of the last repeating unit, R5, but it's only a couple of amino acids. The rest of it seems to be a really stable core. And then I really like your point about, using that. We know that that's a really stable core. Does that change in some of these mutants, or depending on the way that you nucleate the fiber or get the fiber started? And we certainly haven't done that, but it's a great suggestion, Prakash.
And it is also, it might be even more interesting for FABC because it has these linkers which could get involved in the fibril formation to a greater extent. Yeah. So good point, Prakash. To do that.
questions in the chat box from, there's one from Wong. Wong, you can ask directly or I can read it for you.
I already got the answer. Thank
Let's
you.
discuss that. Wong is asking, are there any known functional amyloids from human tissues so far found?
I already have the answer, yeah.
But let's record the answer, for others.
Yeah, but it's
Can we
the
discuss that? Yeah.
Yeah, the answer that he got are great. There are many of them, and more seem to be, sort of pointed at all the time. The PMAL amyloids, I think, were the first ones in human cells. The PMAL lays down a scaffold for melanin formation, and these hormones, these peptide hormones that are stored in these secretory granules, just some brilliant work from several groups have shown that those are apparently stored in an amyloid-like form, and then they can be peeled off of the end of that amyloid to go be active as a peptide hormone. And so there you have, sort of combining a lot of different things, the stability and the ability of those fibers. We think of them as bricks, but they're not. They're dynamic and sometimes dynamic enough and can be regulated enough that that's how you get certain amounts of peptide hormones secreted. So
they are more reversible there? Are they more reversible than the A beta or something?
Some of those peptide hormones are apparently more reversible under certain conditions, and that's really the important point. And I think drives home the point about them being functional, right? They want to be have inputs, whether or not you're going to form the amyloid or maybe break out of the amyloid and go do something else. You want that to be controllable. You want that to be under some sort of directive from the environment and or the cell, and I think those are good examples of them. So under certain conditions, yes, they're much more reversible.
So in that case, does your bacteria do something good or bad to those?
Yeah, no, that's a good question. We've never looked. I don't know. Yeah, it's a good question.
Okay, thank you.
Carmelo, ask a question.
Hi, Daniel.
Hi, Carmelo.
I have a simple question for you. It's known that the sequence glycine XX glycine sequence is very important in the aggregation in membrane, like E beta and the form pore. Did you check if this sequence glycine XX has any influence in the aggregation of your proteins? Because I test in my lab some small peptide with this sequence changing the XX amino acid. And in water, we obtain different results about fibril formation. Do you check about this sequence in your proteins?
That's a good point.
If influence the kinetic of formation of your fibril?
We haven't checked that. I think the GXXG you mentioned, is that the glycophorin dimerization motif that you're alluding to, which of course promotes helix interactions in the membrane.
Yes.
But it would obviously be a very different context for a beta sheet amyloid protein. I mean, as Matt mentioned, the glycines and also aspartates, they play an important role, but I think that's more in the sense of gatekeeping, of slowing down the aggregation to some extent. I'm just looking at the sequence of FAP-C or CSGA, and I'm not sure. I'm trying to find out if they actually whether they have these GXXG motifs. I mean, they have lots of, for example, multiple glycines next to each other, but I don't think that we have actual GXXG motifs there. So, and it would be a different context. So I think that I certainly interpret it in the light of Matt's gatekeeper interpretation. which I think is an important way of just regulating the speed of aggregation or the extent of aggregation.
Okay, thanks.
Thanks. Sorry, I couldn't be more specific.
Anup, go ahead.
Hi, I have a question for both. Great talks, Matt and Daniel. I don't know if there is a concrete, clear answer for this yet. I was talking about this to Rams recently, that in the same system, like in the human body, there could be a disease amyloid and functional amyloid that coexist. But generally, it's like naturally kind of separated. One would not imagine how a bacterial amyloid could affect something in the neurons. But Matt did show some examples today. So what do you think actually causes this interaction? Now that, see, the sequence thing, people have been trying over the years if there are anything common across all the amyloid forming proteins, but it's not necessarily true that there is really any sequence similarities. But still, there are some functional amyloids that are able to interact with disease amyloids. They can cross-seed or prevent aggregation. So what do you think about this? How do they really interact? It's just about the structure, or do you have any other ideas?
I don't know, Matt. You've done a lot of great work on identifying motifs. Do you have any thoughts on this?
Yeah, yeah, a little bit. So you know, one we really wanted it to be. We were really thinking that there was, you know, a particular sequence, you know, that would be driving this sort of cross-seeding or acceleration or, you know, interaction. And more and more we're thinking, you know, back to some of the, you know, work that Raquez and everybody did early on to define these small oligomers. And we think maybe there is some oligomeric state, and maybe that results in a charge state or an occupation state of that particular protein that then allows some sort of early interaction. Maybe we know that CSGA, for example, if just consider CSGA and alpha-synuclein, we know that those two proteins will come together. So if you mix CSGA with alpha-synuclein fibrils, you'll get CSGA to bind to those fibrils. There will be a nice interaction there. And so maybe something that's happening is that they can serve as sort of nucleation points or concentration points or points where you can sort of recruit multiple proteins to the same area. And then alpha-synuclein maybe on its own, you know, gets into an amyloid, but you've provided this small oligomeric template that helps recruit those multiple alpha-synucleins together. You know, I'm wishy-washy on this answer because we don't have a clear one right now. We've certainly eliminated a lot of different things, but we don't have that. Again, we don't have that, you know, definitive answer.
I'd just like to add to that it's unfortunately we find it very difficult to study early stages of aggregation of CSGA because of its inherent tendency to form these also initial state aggregates. You can try to do, for example, 2-photon spectroscopy where you, if you want to do cross-seeding between two different species, so you can see if they co-migrate. But given CSGA, at least in our hands, tendency to aggregate at an early stage, I think it's going to be difficult to get some clear answers unless you work very carefully.
Bina, you're next.
Hi.
Hi,
like
Bina.
this idea of taking the M values from denaturation to the aggregation. I really like that concept. Thanks. My question is regarding your deletion of repeats in FAP protein, and I'm a little surprised that you've taken the entire. It's a significant length of amino acids, and you still see the protein aggregate because the native state of that protein would be pretty much affected, isn't it? Because you've taken a few 10 of residues. Is that the reason why you have fragmentation? Because it's now in a state that it's partially folded. It's not having the entire sequence to form a structure.
Well, it's a good point, because of course in a normal globular protein you would expect that taking out something which is an integral part of the core of the protein would rip the soul out of your protein and prevent it from folding properly. But when you've got a modular folding, as we think is the case with these functional amyloids, then it's a bit like just removing one of multiple units, and the rest should get on with it themselves. Of course, it's a very simplistic view, because there will be interactions not just within but between the individual folding units, because they will be stacking on top of each other. So it's true that you may affect this because you add a new interface, whereas before you had like R1, R2, R3. If you remove R2, then you force R1 and R3 to come together, and that can be a problem. But surprisingly, I mean, that the protein survives removal of one repeat relatively well. So I think it adds credence to the idea that you have individual folding units stacking on top of each other, and you only really tip it when you remove all of them, because then you have no strongly favored minimal folding unit. You just have the flotsam and jetsam, what's left and what has been put into this evolutionarily to perhaps just we see the linker regions, which are left as being not necessarily the icing on the cake, but they are perhaps the more a bit more of a sort of a cover that regulates the hydrophilicity of the proteins. So the fact that they can form amyloid is perhaps a little incidental in that regard.
Okay, could I ask one more, just sequence analysis kind of question? So now that we have a significant number of functional amyloids, has there been any sequence analysis to see how regions responsible for amyloid formation are different from the ones that are present in the pathogenic amyloids? Is there some physicochemical properties of these stretches different between the functional amyloids versus pathogenic amyloids forming proteins?
Yeah, sure. A very, very appropriate question. There is, but what it is perhaps a little difficult to say. Certainly when we've run our functional amyloids through programs like Agroscan, Segregator, those kinds of things, which are used to identify aggregation-prone residues in pathological amyloid, they don't come up very clearly with good hits. Again, I'm sure Matt can nuance this very well, but it's certainly my impression that the functional amyloid sequences don't show up as clearly as amyloidogenic, at least when I've looked at CSGA and FABC.
Yeah, they don't, but I would say that they are almost always predicted to be intrinsically disordered and have some tendency, would be predicted to have some tendency to form an aggregate or an amyloid. So I do think that there's a little bit from the sequence there. And then just to expand a little bit on Daniel's answer, I think the comparison sequence-wise, between pathogenic amyloids has confounded people, for a long time. Just to, I think the idea, 20 or 30 years ago was there must be a consensus sequence which is leading to these fibers, which we can all appreciate look so much alike. And yet that has not necessarily been the case. As we know, many different protein sequences and conformations of those sequences can end up in that amyloid fold. And I don't think that excludes the functional amyloids. I think Daniel's work on the, showing the differences in the primary and secondary nucleation tendencies of these is probably the best differentiator right now that I know of the of like a hallmark of the functional amyloids versus the pathogenic amyloids.
Also,
thank you.
Thanks.
So I wanted to ask you a question on, maybe this is for Matt. Great talks, both of you. You both give great talks. I like it very much. And the synuclein Curli interaction, I know going back to the thesis, Brandon Rotolo's student's thesis committee presentation. So we know that the LPS or membrane components are going to speed up the aggregation of Curli to form fibers, right? Am I correct on that?
Yes.
So what would happen, synuclein also forms like oligomers and most likely helical oligomers. Can you comment on that in the presence of membrane? How would the interaction between Curli and synuclein?
Yeah, it's a great. That's a great question. We haven't done that yet, Rams, but we want to. We keep talking about it. We just haven't gotten around to it. It's a great question.
Doing it any biological significance?
Well, certainly. I mean, LPS. You know, as we've talked about, you know, LPS is a really charged molecule, right? And it is obviously where the Curli proteins. So a protein that we haven't mentioned too much is the CSGB protein, which is, we call it the nucleator protein in the E. coli system. That protein, it's an amylogenic protein itself, but it's got one end of it which sticks into the LPS, or at least sticks very tightly to the LPS, prevents itself from getting into a multimer, an amyloid-like multimer, but it does fold into a single subunit cross beta strand structure. And we think that very clearly, I think we've shown and other groups have shown, that very clearly serves as a template for CSGA amyloid formation. Well, we would love to know. And in that case, LPS is really a facilitator for CSGB function. If you put LPS there, you get this nice interaction with the CSGB, tethers it, and then allows that template to form more efficiently. And so, yes, we would love to know what the collective interaction there is between an alpha-synuclein and the LPS and the Curli-like proteins. We know that LPS can affect at least the CSG proteins. We would love to know what it does to the alpha-synuclein or other human proteins.
So from your data, we know that monomer-monomer interaction is the main driving force for synuclein aggregation in the presence of Curli. So in the presence of LPS, Curli is going to be fibers very quickly. So would that mean, we know that synuclein is going to go to alpha helical structure. Would that mean that there won't be any crosstalk, any cross-seeding, or any Curli-assisted aggregation?
Yeah, it's a good, we don't know. Yeah, I mean, yeah, it would be one possibility. Yeah.
And one thing is that alpha-synuclein is tricky because small amounts of detergent can actually stimulate fibrillation. If it's below the critical micelle concentration, it can encourage the proteins to essentially assemble around the bonding micelle, and so you encourage aggregation. You can actually get a beautifully reproducible and controllable aggregation profile for alpha-synuclein by just having a little bit of SDS presence. It's like 0.5 millimolar. You don't get bona fide long fibrils. They become more curly because they are probably plasticized by SDS, but they certainly are stimulated to form aggregates very, very nicely by SDS. And I think LPS has the same tendency as well.
But we know that anionic lipids would promote the helical structure formation and oligomer formation in membrane for synuclein. So maybe the curvature is playing a role as opposed to anionic charge per se.
Astrid knows so much about protein.
Oh, Astrid, go ahead, Astrid. Yeah.
Yes, just a comment to Daniel's comment about the role of SDS, that yes, detergents in very small amounts can definitely enhance the aggregation and amyloid formation. For instance, also A-beta, which I have studied at some early stage. And I'm sure we will find it the same thing if we would go for other detergents as well. So you both have the secondary structure transition to beta sheet, and you have the big, you could say, aggregates formed, which will be, I think, some kind of, you could say, mimic of membrane activities here.
All right, it looks like there are no other questions. Thank you both very much for outstanding presentations and great Q&A sessions. Sorry for the Zoom mess. We'll fix it next week.
No
Thank
problem.
you all very much. Nice seeing you. Astrid, can you hang around? Griesinger, can you hang around